Hi everyone, thank you for coming. I'm going to be talking about the Elasmobranch and the slough. Estuaries are important coastal habitats that provide numerous benefits for humans, including acting as a buffer from floods, winds, storms, and waves. They also filter numerous toxins that flow through the watershed and provide resources such as shellfish, finfish, and types of vegetation. Estuaries also provide numerous benefits for animals, including habitat complexity, allowing animals to avoid larger predators, an abundance of food due to the high nutrient content, and often warmer, shallower habitat that enables animals to grow at a faster rate. All of this leads estuaries to act as suitable nursery habitats for a lot of different species of fish and invertebrates, such as endangered species and commercial species, including Chinook salmon, Dungeness crab, and English sole. Elkhorn Slough is an estuarine embayment that drains directly into the Monterey Bay at the head of the Monterey Submarine Canyon. Although many of the sloughs around California are more fresh, this is a mainly marine slough, with the main sources of fresh water today being rainfall and agricultural runoff, and occasional input more in the wet season from Old Salinas River Channel, Carneros Creek, and Morro Coho Slough. Elkhorn Slough is approximately 11.5 kilometers long with an average depth of 1.4 meters and a maximum depth of 7.5 meters at the entrance of the slough by the harbor. The structure of the slough is a winding main channel with several tidal creeks that come off of the channel, and salt marsh habitat in green and mudflat habitat in brown lining the sides of the channel, with most of the salt marsh occurring more in the mid and lower slough. Like many of the estuaries around the world, Elkhorn Slough has undergone several changes over the last couple centuries, and since the late 1800s, agriculture, ranching, and increased coastal development have altered the structure and natural evolution of Elkhorn Slough. The diking and draining of lands for agriculture and ranching, the building of the Southern Pacific Railroad, and evaporative salt ponds have removed a lot of wetland habitat from tidal exchange. Additionally, in 1908, the Salinas River mouth was relocated due to storms from north of Elkhorn Slough to south of Elkhorn Slough, drastically reducing the amount of fresh water and sediment input into the slough. However, the most significant change occurred in 1946 and 1947 with the building of Moss Landing Harbor. This opened Elkhorn Slough directly to Monterey Bay, increasing the tidal influence and incursion into the slough and causing it to become more marine with stronger current. Further failure of levees and dikes increased the tidal prism of the slough, causing it to become more erosional and change from a depositional to erosional habitat. This has led to a decrease in healthy salt marsh habitat in the green and an increase in mudflat. Overall, high-quality salt marsh area has declined to 23% of its original value since 1870, being largely converted into this mudflat. Now moving on to Elasmobranchs. Much of what we know about the Elasmobranchs in Elkhorn Slough comes from the shark derbies that were initiated in 1946 by local rod and gun clubs. These occurred 1 to 2 times a year in May to August and lasted until 1995. Each day, 300 to 600 fishermen would participate in the derbies, catching approximately 100 animals. And in 1951, Earl S. Harold from the California Academy of Science began to start recording data from these derbies and publishing information about the species compositions and the Elasmobranchs in the slough. In 1971, Moss Landing Marine Lab students began to participate in this process of helping out with the derbies and would weigh and measure animals collected and also collect opportunistic samples for diet, reproduction, and aging studies. In 1988, Aaron King initiated a tag and release program as well that lasted until the end of the derbies. From these studies, we found that there are 7 species of Elasmobranchs that have been commonly caught throughout the derbies. For the sharks, this includes the leopard shark, the brown smoothhound, gray smoothhound, and for the rays, we have bat rays, shovel-nosed guitarfish, thornback rays, and brown stingrays. And these are all caught along the California coast with different ranges. So several studies or several theses have been published based off of this derby data, including the age and growth of the bat ray, brown and gray smoothhounds, and the leopard shark, the trophic ecology of all of the above species of Elasmobranchs, fishes of Elkhorn Slough, and more specific studies on the gray smoothhounds and leopard sharks. And then more recently, there have been tagging studies looking at the movements of female leopard sharks and then male and female leopard sharks. So although you can see that a lot of studies have already been conducted on the Elasmobranchs in the slough, most of these studies have focused only on one species or on one small spatial distribution. So for my study, I wanted to get more of an overall standardized picture of what was going on in the slough now and over multiple locations. So for the next 40-ish minutes, this will be the structure of the talk, where I'll start with part 1, the current Elasmobranch assemblage, and go through the objectives, methods, results, discussion, and summary. You'll think it's over, but then we'll have part 2, the historical Elasmobranch assemblage, and I'll go through all of these sections again, but it'll be much shorter. And then I'll get to some conclusions. So let's start with part 1, the current Elasmobranch assemblage. And Helena's in the back. Celebrity. My objectives for part 1 were to determine which Elasmobranch species are common, describe the spatial and temporal distribution of common species by sex and life stage, and to determine how variability in environmental and physical characteristics influence the distribution patterns of Elasmobranchs in the slough. I want you to pay attention to the colors and the numbers in these boxes, as the slides are going to have these boxes in the corners of each to represent what objective I'm addressing. So for my methods, I went into the field from April 2015 to August 2016, sampling approximately 8 days per month, though due to weather, there were a couple days that were cut off here and there. And at the end of my study, gill netting was more restricted due to some permitting issues. And I used longlines baited with squid that soaked for 3.5 hours at a time, and gill nets that were different sizes based on where I was fishing, that were 5- and 7-inch stretch mesh, and that soaked for 20 minutes at a time. Most of the sampling was done off of a whaler, a small boat. In some of the areas, we did swim out the gear off this bridge. While out there, we collected information on temperature, salinity, dissolved oxygen, depth, and Secchi depth using YSI, Secchi disc, and depth sounder. Sex size and maturity for each animal that was caught, and following my IACUC protocol, which involved catching and releasing most of the animals except a subset of thornback rays that were dissected for life history studies. Also, I collected GPS locations on the nets and the lines and externally tagged the animals. So just for a picture of what happened each day, we'd start after soaking the net or the line, we'd pull up the gear, remove the animal from the hook or the net, measure and sex the animal, tag them with either a Peterson disc tag for the rays or a Floy tag for the sharks, and then release them. Here are my sampling locations overall. for the gillnet and the longline. I divided up the sections into lower, mid, upper slough, and Esner. And you'll see these numbers later, but I'll mostly refer to them this way. I had initially wanted to sample throughout the slough, but ended up focusing on the mid and upper slough due to there being a high density of otters and kayakers in the lower slough, as well as high current, and then very shallow depths in the upper slough, making it hard to find somewhere to lay out the entire net. And this Esner area that I circled here is a more recently restored habitat from the 1980s that's now managed by the Department of Fish and Wildlife and monitored by NOAA's National Estuarine Research Reserve Program. And this area was found to be important for certain species of Elasmobranchs in earlier studies. And so I sampled in one area here that other students had sampled in before. I'm not going to go over all of these, but I wanted to just show you the analyses that were conducted for Objective 2, describing the spatial and temporal distribution, and you'll see the results later on. And then for Part 3, looking at the physical and environmental variables, I focused in this presentation just on the canonical correspondence analysis, which is a multivariate analysis, just so I'd have enough time to explain. So now I'm going to move on to results. Overall, 6 out of the 7 mentioned species were caught. Bat rays were most common, with a total of 311 females and 207 males caught. Leopard sharks were next most common, with 154 females and 134 males. And then thornback rays with 134 females and 17 males. So I'm going to focus my analyses on these 3 species for the rest of the study, as the other species, the smoothhounds and the shovel-nosed guitarfish, were very rarely caught. The results from the gillnet and longline were pretty different. Although the primary species were caught in both of these methods, the gillnet was much better at catching the sharks. So all of the smoothhounds were in there, as well as most of the leopard sharks. And it also caught a portion of the bat rays. And leopard sharks are yellow, bat rays are pink. However, the longline was much better at catching the bat rays, and also the thornback rays in blue. Other interesting catch from my gill nets and from some otter trawls that I did as part of the study as well included some fun invertebrates, such as the navinax and the sea hares. Different species of fishes, including rockfish and flatfish. And even this yellowtail amberjack that is a more of a southern species, but this study occurred during an El Nino year. And so this fish was most likely attracted to the slough by the warmer El Nino waters. And then other unintended catch included some slough artifacts and floating dead fish and some parasites that one of my interns looked into for the leopard sharks and bat rays. Okay, so now for looking at spatial and temporal trends. This is a day when our site got really flooded, so in the winter. So for my analyses, I divided up my seasons into wet and dry seasons based on the conditions of the year. I did want to mention that this dry season of 2016 was a little shorter than the dry season of 2015, as I didn't carry out the November or September and October. And then my zones were divided into lower, mid, and upper slough, as well as Esner. And as I mentioned, although there was a little bit of sampling in the lower slough, for the longline, I'm not including those in the analyses, which is why you might just see 2, 3, and 4 later, which are 2, 3, and 4. And then for my species, I'm going to focused, as I mentioned, on the 3 common species, so leopard sharks, bat rays, and thornback rays, with the leopard sharks coming from the Gill net data, and the bat rays and thornback rays coming from the Longline data. Okay. So looking at this plot, this is the overall average abundance of each species, the leopard sharks, bat rays, and thornback rays, over the course of the sampling period. You'll see that all 3 species follow a similar trend of being more common earlier on in the spring and summer, between April and September, and then starting to decrease in abundance around October, November, and then returning again to the slough as we get to February, March, or April, depending on the species. This pattern of seasonal migration is commonly seen in these species and other Elasmobranch species in other bays and estuaries in California. Now I'm going to focus on each individual species and talk about the pattern seen for each species on its own. Leopard sharks were primarily caught in the mid-slough, so 81% of all of the leopard sharks corrected for catch. Oh, I didn't mention catch per unit effort earlier, but for my catch data, I standardized all of it for the Gill net by, so it's total catch by the number of feet of the net by hour, and then for long lines, it's total number by hook by hour. So as I mentioned, most of the leopard sharks were caught in the mid-slough, which is in the blue outline over here. The larger circles indicate higher catch. And then also the leopard sharks were more commonly caught in the dry seasons. So here we have dry season, wet season, dry season. To look at this statistically, I conducted a Two-Way Anova by season and zone, and found that there was a significant interaction between zone and season. This is because the overall trend, if you look at the plot, is that there were more leopard sharks in the mid-slough and then less in the upper slough and Esner. However, there was one period in the dry season of 2016 where there are a lot of leopard sharks caught in Esner. And so the trend was not consistent by zone. There. And the sex ratio of 1.12 females to 1 male was not significantly different from 1 to 1. While the size distribution between males and females was significantly different, however, the p-value was 0.03, so pretty close to 0.05. The main difference here was that while there was a narrower range of where females were common between 115 and 130 centimeters, males were more common at the larger end of their range, kind of between 110 and 140. And then the black line, I wanted to mention, it marks the size at maturity. So although I did take some maturity data on the males, I ended up using the published length at maturity data to determine between mature and immature. And then for the long lines, I did not catch a lot of leopard sharks, but you will notice that most of the ones I caught were on the smaller range. I conducted just a one-way ANOVA on season to look at whether or not size was significantly different because sample size in Esner was too small to look at zone. Season was not significantly different, though there were slightly bigger leopard sharks in the mid-slough in the wet season. I'm going to go through this plot in detail. So I just want to show you that on the bottom, this x-axis here, we have month. So starting from April 2015 going to August 2016. I've divided it up into dry season, wet season, and dry season to help you look at the seasonality of this. And then for each month, we have zones 2, 3, and 4, which are the mid-slough, upper slough, and Esner. So you see that there are 3 bars here, and each bar corresponds to April 2015 in zone 2, 3, and 4. And within each bar, we have immature females, immature males, and mature males. As I mentioned, most of the leopard sharks were caught in the mid-slough, so you can just mostly focus on that. So one thing interesting from this plot is that all of the sex and life stages are pretty well represented, which you'll see is different in the other species. And also, I wanted to point out the pattern with mature female leopard sharks versus mature male. So mature females in the dark pink remained in Elkhorn Slough throughout the sampling period and in the wet season as well, and didn't really change too much in abundance either. However, the mature male leopard sharks, while they made up around 46 to 79% of the catch per unit effort during this dry season, and 44 to 61 of the second dry season, they only made up 0 to 30% of the population between October and February during the wet season. Another study by Andrea Launer also found in her gillnet surveys that sex ratio of females to males increased in the fall and winter. Although male leopard sharks may be more susceptible to environmental changes, it's also possible that the temporary increase of the males is timed with mating, which likely takes place after females give birth in April and May. Now moving on to bat rays. Most of the bat rays that were caught were immature. 96% of all of the females and 84% of the males were immature. I divided up this plot into mature bat rays versus immature bat rays. And as you can see, spatially, these immature rays were caught throughout Elkhorn Slough, whereas mature rays were only in the mid-slough. And then seasonally, immature rays were caught during all seasons, and mature rays were only caught in the dry seasons. For the Two-Way Anova, both zone and season were found to significantly influence the catch per unit effort, with the mid-slough being where most of the bat rays were caught, just like the leopard sharks, and the dry seasons also having higher abundance of bat rays. The sex ratio of 1.58 females to 1 male was significantly different from 1 to 1, though most of these females were immature. And the size distribution was significantly different between males and females, though the p-value here was also close to 0.05. For the gillnet, the Kolmogorov-Smirnov test here was conducted for the Longline data, and so the main difference you can see from the distributions are that females got to a larger size overall compared to males, but there were very few individuals of these. But the main difference in the distribution falls here on the larger end of female size, where males had more of a condensed size distribution. The results for the Two-Way Anova indicated that season and zone were both significantly different for mean size, with bat rays being larger in the mid-slough, likely due to this being where the mature animals were, and also in the dry seasons, as this is also when the mature bat rays were present. For this monthly distribution plot, you'll see here that there's much more of the light pink and the light blue because these are almost all immature individuals. And it was during June through August and July and August here that you see the mature females coinciding with the mature males. Martin and Caillet similarly found in 1988 that mature bat rays entered the slough in May to give birth and mate and departed by late September, while another study in Humboldt Bay found that large bat rays were most common in June and July, with a subsequent increase in immature rays in September. So the presence of these mature individuals around this range indicates that bat rays are giving birth in July and August. The seasonal presence of reproductively mature and active individuals is common in other species of sharks, which prefer deeper, colder waters during the non-reproductive season, but migrate near shore in the summer to give birth and or reproduce. Now onto thornback rays. All of the thornback rays that were caught were mature, and all of this, so a lot has been done on leopard sharks and bat rays in previous studies, but everything on the thornback rays is pretty new, as they're more of a new species in the slough. And I found the patterns with the rays, these rays, to be very interesting. So spatially, males were only caught in the mid-slough, and I think there was one individual in the lower slough, while females were caught throughout Elkhorn Slough. And then seasonally, they were both completely absent in the wet season, but you'll see more of a fine-scale picture in the monthly plot. The Two-Way Anova indicated that there was a significant season and zone interaction due to there being generally similar, due to there being a lot of thornback rays in Esner during the first dry season, and then very few, only 2 individuals caught in the second dry season. So this pattern became pretty inconsistent. The sex ratio was 9.2 females to 1 male and was significantly different from 1 to 1. The size distribution was also significantly different between males and females, with there just being a much broader range of females fitting more of a normal distribution, while there was a very small size range for males. For the Two-Way Anova, zone and season were both significantly influencing the size of thornback rays. As I mentioned, the males were only in the mid-slough and they were smaller, so they brought down the overall size. And overall, females seemed to be larger in Esner compared to the upper slough and the mid-slough. The monthly distribution of thornback rays was pretty interesting. So while the leopard sharks and the bat rays consistently preferred zone 2 or the mid-slough, the thornback rays seemed to shift their preference throughout the season. Also, in comparison to the bat rays and thornback rays, these rays actually use some of the other habitats more, the upper slough and Esner. And these stars indicate where the thornback rays were most abundant in the dry season of 2015 here, when they're in zones 3 and 4. So at the beginning of this season, they were most frequently using the upper slough and Esner, which is shallower, warmer habitat. And then this seemed to shift around July as they began to use other habitats more. In 2016, the pattern was a little bit different, but they did preferentially use the upper slough for a period of time in June and July, which is where the stars are in Esner here. Additionally, the males became more abundant at the end of the dry seasons, August, September, and July, August. So I was trying to explain this pattern, and there was another study that looked at brown stingrays in Southern California that found a similar pattern of mature female rays using shallower, warmer habitats at the beginning of the season, and they attributed this to them wanting to use these warmer habitats early in gestation to decrease the amount of time that embryos were gestating, or perhaps to increase the growth rate of the embryos. So this was similarly seen in the thornback rays at the beginning of 2015. And for my life history studies, so for the rays that were dissected, all of them were pregnant, and throughout the season I was able to see this phase of development shift from the animals being undeveloped to slightly developed to fully developed. And so they are gestating in the slough. Another similarity seen with the Jurek and Lowe Southern California study was that neonates were not observed. So in that study, they proposed that the habitat was a maternity ward rather than being a nursery, where the round stingrays were just using the habitat during gestation but not giving birth. And because no neonates were caught, it's possible that this is happening here as well. However, I did see very well-developed thornback rays, and so it's also possible that I just wasn't able to catch them based on the gear and the locations that I was sampling in. Another interesting observation was instance of mating behavior. So in this picture, we have the large one being a female thornback that was caught on the Longline, and I pulled up the line to find 2 males attached to it. This biting of the pectoral fins is classic Elasmobranch mating behavior, and so it indicates that although I didn't see the actual physical act of mating, that thornback rays may be mating around this time. And this occurred in both years, once in late August and once in early September. So moving on to a brief description about the environment in Elkhorn Slough. I wanted to compare Esner and the lower slough. This data is taken from the National Estuary Research Reserve Monitoring Program. When you compare Esner, which is less tidally influenced, with the lower slough, which is much closer to the Monterey Bay, you see there's much more fluctuation in the values compared to the lower slough. And then in general, temperatures are higher in the summer with higher salinities, lower dissolved oxygen, lower temperatures in the winter, lower salinities, higher dissolved oxygen, and then back to the same for the dry seasons again. And to compare the lower slough to Monterey Bay, temperatures are generally higher than Monterey Bay, except for a brief period in the wet season. Salinities are generally similar or lower after the heavy rain season. And then dissolved oxygen is generally similar except during some periods in the summer. And same patterns with Esner except with to a greater degree. Now I'm going to move on to relationships between the assemblage and environmental variables. This plot is a little bit confusing, so I'm going to spend some time on it. This plot is for a canonical correspondence analysis, which determines the structure between 2 sets of variables, the species and the environmental and physical variables, and looks for a relationship between these 2. So in blue, we have the different species groupings. I took out the thornback rays as rare species can influence the results. In black, we have the discrete variables, so the month, zone, season. And then in orange, we have the continuous numerical variables, such as temperature, salinity, depth. When you see one of the arrows pointing towards a certain direction, it indicates that this is a higher value. So pointing to the left means on this side, we have higher temperature. Meanwhile, there's no arrow on the other side, but really it's indicating that on the other direct opposite side, we have lower temperatures. So high temperature, low temperature, high salinity, low salinity, etc. And then species that are closer with each other are more similar to each other. And the analysis, we end up with these 2 axes, which are trying to maximize the variance that's explained between all of these different species groupings. I'm going to move on to the results now. So immature bat rays are on the negative end of this axis. Things that are associated with these immature bat rays include the upper slough, so they're more common in the upper slough compared to other species, higher temperatures, higher salinities, and then July and August. Meanwhile, leopard sharks are all pretty clustered together and fall a little bit more towards the right side. These were associated with the wet season, the mid-slough, and then these lower values of temperature, higher dissolved oxygen, and Secchi depth. For the Longline CCA, we have female thornback rays falling on the left side and highly associated with Esner in May and June, and the upper slough as well. And there were higher temperatures and salinities here. Male thornback rays and mature bat rays all were more common, as mentioned, in the mid-slough in the dry season of 2016 and under greater depths and secchi depths. And then the immature bat rays fall pretty close to the middle. Usually things towards the middle are not highly associated with anything, and immature bat rays were caught throughout the seasons and zones, so this makes sense. So now I'm going to look at each species patterns with environmental variables. Although we looked at some of these patterns with different elasmobranchs and variables, I wanted to look at just the leopard shark overall and some of the correlations with temperature, salinity, and dissolved oxygen that have been suggested to influence seasonal migration in other estuaries. So I just plotted the catch per unit effort against these different variables in both Esner and the lower slough. These green lines indicate thresholds at which elasmobranchs were found to leave estuaries and other systems. Temperature was found to correlate the best with, leopard sharks were found to correlate best with temperature, increasing during the warmer temperature periods, decreasing during the lower temperature periods, while they didn't correspond as well with salinity or dissolved oxygen. Previous studies, such as by Aaron Carlisle, found that dissolved oxygen was important in influencing the movements of leopard sharks out of Esner. However, because there were so few sharks caught in Esner, I wasn't really able to look at this pattern. The dissolved oxygen values did get pretty low there. For bat rays, we again see a similar trend with temperature. Another interesting pattern that was seen was in the wet season. We had 2 high rain months, January and March. This decreased the salinity of the slough, causing some of these lower peaks, and also corresponded with low abundances of bat rays. Now, this could be just attributed with bat rays not having come back to the slough yet. However, in February, there was a lot less rain here, and their abundance temporarily increased. And so it's possible that bat rays are much more susceptible to changes in salinity compared with leopard sharks. And they have been found in physiological studies to be highly influenced by temperature and salinity as it increases the rate of oxygen consumption. There were less clear relationships for the Thornback data. While they did decrease during the lower temperatures, they seemed to decrease quite a lot sooner than the temperature started changing. And so likely these relationships were more related to the reproduction that I was talking about. So in summary, I'm going to go over each objective and talk about what we found out. Bat rays, leopard sharks, and thornback rays were the most common species. And gear type mattered in determining the species composition. Some explanations for why this might be. The gill net was a little bit more targeted towards certain animals. It relies on an animal of the right shape and size based on the mesh size to swim into the net and has to get caught on the gills. This prevents the animal from swimming out. The rays weren't shaped as well for this gill net and so weren't caught as well. Their gills are on the bottom of their heads and they're like a rounder shape or a flatter rounder shape that wouldn't get caught as easily. For the long lines, I was trying to target a broader range of animals. So I used different hook sizes to try and get some of the smaller ranges and larger ranges that the gill net couldn't catch. However, long lines are only targeted towards hungry animals as they're baited. And so it's possible that some of these animals just weren't hungry or that something about the way the long line was set up. And I did try some otter trawls to catch some of the smaller individuals later on in the season, but was only successful at catching large thornback rays. For objective 2, distribution did differ by species and life stage. This is a summary table of what I found were the common patterns. So leopard sharks, they had a pretty similar distribution by sex and life stage, except for mature females that remained in the slough year-round. Bat rays showed a difference between immature and mature individuals. As I mentioned, immature individuals use the slough year-round and all over the slough. And thornback rays had a slightly different temporal distribution, with males coming in the late summer and only being in the mid-slough as well. So for objective 3, we did see from the analyses that some of these spatial and temporal variables like zone, season, and month were important in influencing the distribution of these species, as well as environmental variables such as temperature, salinity, Secchi depth, and dissolved oxygen. I did want to mention that a lot of these variables are correlated with each other. So this principal components analysis takes all of the variables that you're interested in, so for the environmental variables here, and rather than having to look at these relationships by one variable at a time, it combines all of these variables into one axis so that you can just use this one PC1 axis to explain these different variables that are correlated with each other. Positive values on this PC1 mean higher values of dissolved oxygen, depth, Secchi depth, and lower values of salinity and temperature. And so you can see that a lot of these are either positively or negatively correlated with each other. And then I won't go into detail about the symbols and colors, but they do represent zone and season, and you can see separation here. And so these are also correlated with some of these variables. But in summary, for the temporal distribution, on a seasonal scale, distribution was influenced by environmental conditions such as temperature and salinity, and on a finer scale by parturition and reproduction, as seen in the mature bat rays and thornbacks. Spatially, it's possible that the leopard sharks and bat rays that were more common in the mid-slough were more attracted to the stability of that environment, as there was less environmental change. There's also the possibility that prey distribution played an important role, as mature bat rays and leopard sharks have greater energetic needs, and the larger, more diverse prey are found in the lower and mid-slough where they were common. And then for female thornback rays, their spatial distribution was most likely influenced by the gestation period. Okay, now we're on part 2. For my objectives, I'm going to I wanted to describe how the species composition has changed over time and determine how long-term changes to Elkhorn Slough have influenced the species assemblage. A previous study by Carlisle et al. In 2007 has looked at some of these trends already, but I further plotted some of the data from the derbies and other studies to look at proportional abundance and how it's changed. And then also conducted some linear models and generalized linear models to look at variables that may have influenced the assemblage. So human alteration has led to a great loss of estuarine habitat and function. However, some studies have actually found that there are positive relationships between elasmobranchs and human alteration. This includes for bull sharks, they were found to show site fidelity towards dredged creeks and channels. Round stingrays were found to prefer habitat around thermal outfalls. And leopard sharks and other Southern California species were found to be common in a lot of the human restored sites as compared to other habitats. Here is the proportional species data from the derbies. You'll see pink for the bat rays and yellow for the leopard sharks. These species remained common throughout the period of the derbies. Meanwhile, other species such as the green shovel-nosed guitarfish, blue thornback rays, and gray smoothhounds varied more during this period. Now, one of the relationships that I found most interesting and that the Carlisle et al. Paper brought up was the relationship between shovel-nosed guitarfish and thornback rays, which are pretty similar in terms of the habitats they use and the types of food they consume. And so I looked at the species proportional abundances and how they changed just between these two species. And this shows that shovel-nosed guitarfish seem to have decreased around the same time that thornback rays have increased. This seems to have occurred around the 1970s. And other studies have found that, have confirmed some of these patterns, with bat rays and leopard sharks being consistently common, and there being more shovel-nosed guitarfish earlier on and more thornback rays later on. And these are the studies. These studies do all use different types of gear, mostly gill net, but some have used otter trawl and beach seine, and different spatial and temporal ranges, so just keep that in mind. And so some of the variables that I looked into were habitat change, as I mentioned, the increase in mudflat and decrease in salt marsh, as well as the erosion that has led to an increase in creek width. And then some of the large-scale environmental indices, including Pacific Decadal Oscillation, the Multivariate El Nino Index, the North Pacific Gyre Oscillation, and upwelling. The values that are positive on these axes indicate a positive phase, and negative indicating a negative phase. PDO was suggested by Carlisle's paper to have an influence on the shift from guitarfish to thornback rays. And then other studies, such as Clarn et al. 2010, has found that NPGO and PDO shifts had an important impact on the invertebrate and fish assemblages in San Francisco Bay. To look at this more, I conducted another PCA looking at just species proportional data. As I had mentioned earlier, a PCA is useful in reducing the number of variables that you need to use in your analyses and the ability to look at multiple things at one time. And so PC1 on the bottom here, I'm using that to explain high numbers of thornback rays and low numbers of guitarfish. So positive values on PC1 will be corresponded with more thornback rays. And then I plotted this PC1 axis against the variables that I mentioned earlier and found that relationships were significant by decade, PDO, the El Nino, and mudflat area. All of these relationships, so all of them are positive, and so more thornback rays are associated with time overall, with warmer phases of PDO, which are warmer sea surface temperatures in the coastal area, warmer El Nino indices, and more mudflat habitat. I then created a generalized linear model with just the thornback and shovel-nosed guitarfish data, the proportional abundances, and found that while PDO and mudflat were significant in independent models, the final model following the backward stepping format resulted in only decade as being significant. This indicates that changes are happening on a decadal scale, but because all of these variables were associated with each other and correlated, it's possible that the effective decade is masking some of these other finer scale impacts. And then looking at pairwise comparisons, the 50s and 60s were not significantly different from each other. They're just being mostly dominated by guitarfish. And the 80s and 90s were also not different from each other, but every other pair was. In summary, leopard sharks and bat rays were consistently abundant throughout the Derby period, so all the way from the 50s. There has been a proportional decrease in guitarfish that aligns with an increase in thornback rays. So thinking about some of these influences on the assemblage changes did occur on a decadal scale and are possibly associated with PDO and habitat change. Thornback rays were more associated with this shift from a cool PDO phase to a warm PDO phase or warmer temperatures, and may have been attracted to this and come up into Elkhorn Slough. It's possible that when they began to inhabit the slough, they outcompeted guitarfish, or that they have just had a range shift. So they used to be really common in Southern California and less common up around here. Their northern range is Tomales Bay. However, in more recent years, based on personal conversations, they're much less common in Southern California and now seem to be pretty common in the slough. Another variable that I wasn't able to look at was prey distribution. So the increase in erosion has led to a lot of habitat change, as I mentioned, and a lot of the tidal creeks that were used by Elasmobranch have become more like the main channel and decrease the diversity of invertebrates. In addition, otters have begun to populate Elkhorn Slough greatly, and they are voracious consumers that can eat, 35 otters can eat 400,000 crabs per year. They have been found in studies to have decreased clam biomass in size and also cancer crab biomass more recently. And a shift in diet for leopard sharks has been observed in more recent studies, from clams and crabs to more fishes and fat innkeeper worms. So although most of the elasmobranchs are generalist predators, it's possible that these distributions have influenced the elasmobranch abundances as well. In conclusion, Elkhorn Slough remains important habitat for several species of elasmobranchs, including one new species, the thornback ray. It appears that thornback rays have replaced shovel-nosed guitarfish as being one of the more dominant species in the slough, and that this habitat has become important for reproductive stages of the females and possibly also for mating, which hasn't been previously reported. There was a lot of variability in the data, especially I found Esner really frustrating because the patterns weren't consistent. And studies there have sometimes indicated that animals are common and sometimes not. So more research needs to be done there. I did find that spatial and temporal variables and certain environmental variables appeared to be influencing the distribution of a lot of these species. And more studies need to be done. Ideally, it would be great to use other types of gear that are better at targeting some of the neonates. I think everyone would be excited to have some baby pictures of these animals, and it would be great to see if thornback rays are actually giving birth in the slough, since nobody knows. Trophic studies and tagging studies would also give us a finer scale picture of what elasmobranchs are doing outside of the sampling regimen that I had, and to confirm whether or not animals are consuming different items or being influenced by prey distribution. And then longer-term monitoring is vital to help us see how these patterns vary from different types of years and conditions, and will help us understand how they will respond to continuously changing environmental conditions and habitats. That's it. Okay, so I just want to thank a couple people. I'll start with Dave. Thank you for accepting me into your lab, even though I didn't have previous shark experience, and helping me develop a project that allowed me to spend so much time in the field, and just for always being supportive during my sometimes frustrating experiences out there. Scott, I have this classy picture of you and a picture that Stephen gave me of you. Thank you for always having your door open to us, and just for your general interest and dedication in each student, even though you have a lot of students and a lot of workload, but you always were there for us and provided really useful, simple, direct suggestions. Kenneth, I know we didn't spend a whole lot of time together for this project, but you had a lot of expertise on the slough and on the chemical side of things that I didn't really understand. So thank you for your advice and for reading it to make sure that it was appropriate. And I think I've been really lucky to come here while you were still here and teaching. You are great to the students and have always had your door open as well. I don't know if Joe is here, but Joe, you didn't know what you were signing up for when I first asked you for help. I just wanted some advice, but you ended up teaching me all of the stats, all the multivariate stats that I know. And thank you for your thoroughness and your patience with me. Thank you to JD, who's not here, and Daxon, who is. You made me part of the Small Boats family. You taught me a lot about really cool things that I can now brag about, like driving a boat and tying knots, and just how to be good out in the water. So thank you for that and for always being there when I came back from a hard sampling day. Thank you to the lab for, I think all of you came out with me at least a couple times to help. So thank you for your help and for just being a funny and crazy bunch of people that brought me out of my shell a little bit. I had a lot of volunteers, so look for yourself if you helped me. And I couldn't have done my thesis without you guys. I was mostly the captain, and you guys did most of the work. So thank you for all of your physical labor and your conversation, and just for making the experience better for me. And here are all of your names. Thank you to my family. This is the best picture I have of us. It's really, really cute. Thank you for supporting me and not bugging me about finishing until I got towards the end, and just being there and caring about what I do. Thank you to the squad or the lunch bunch for always being down for lemongrass, and just for being there for me. And to Wes, who went to get the lemongrass, thank you for encouraging me to finish so that I could move on to better things and have fun in my life again. And then other people I didn't have specific photos for. Thank you to Aaron Carlisle and Brent Hughes for their help on the historical aspect. Thank you to Rick Kavitek for teaching me GIS and lending me a GPS unit. To John Haskins, Charlie Endress, and Kirsten Wasson and Esner for helping me with various aspects of data or help in the slough. Dr. Richard Starr, Rick isn't here either, but he helped me a lot with permits and just with other questions that I had. He also helped me build my long lines, which was really important. Thank you to Andrea and Kelly, who were the shark people at the time that I started at the lab, and that encouraged me to work out in the slough as well. Thanks, as I said, JD, Jackson, and also Brian Ackerman. Thanks to James, Billy, Chris, and Gary for just always being around to help with whatever was going on or going wrong. The IT department, library staff, and front desk for all of their work as well. And then Jocelyn, Kathleen, and Jane Fusek for hiring me in the museum, which supported me for most of my time here, and let me play around in there and find interesting things, and then all of my funding sources. That's it.
Questions for Kat?
Yeah.
So I was happy to see that you pointed out that otters have changed the prey distribution in the slough and probably changed availability of prey for elasmobranchs. And I was wondering if you think that direct predation by sea otters on elasmobranchs, as sea otters have increased, has influenced the assemblage or distribution of elasmobranchs in the slough.
Yeah, so there was, I wanted to put a picture of this in, but it wasn't mine. There was a brief period of time where the otters were eating the thornback rays, not even the small ones, but the large ones. I heard that it hasn't been happening as recently, but definitely direct predation will have an influence. You would probably know better than I do about what their diet composition looks like, or Sarah, or whoever's working on that. To my knowledge, it isn't a big proportion of their diet, so I don't know that it would change like the overall abundance or the health of the species itself, but maybe as prey dwindles, they'll start turning to other things like the small, the small sharks. So, yeah. Ken?
Okay. You mentioned the importance of long-term monitoring, and you sort of point towards prey distribution as being important in terms of understanding the abundance of these species in your location. What prey item, if you could start a monitoring program, what prey items would you be looking at on a routine basis?
I probably, well, for the sharks, it would be the animals that they consume. I don't know too much about what might be important for other species, but so the Washington and gaper clams are the ones that were found to decrease in size and biomass because of the otters. Those seem to be a highly prized prey item, so tracking where they are and how that changes would probably be a pretty good indicator if the top most popular species is going away. The crabs are definitely also really important, the crabs and the innkeeper worms for both otters and the leopard sharks and some of the larger bat rays, maybe. So I think they're pretty hard to monitor, yeah. Which is maybe why it's not been done so far. But being able to monitor that would also be important. Jan?
So you mentioned that you didn't think it was a nursery for elasmobranchs, and certainly it is, at least in the past, for a lot of other fishes. So even though you didn't catch any, or very many because of the gear, can you talk about a little bit why you think it's not a nursery, and yeah, why? What's the rationale for it? Anyway.
I don't know that it's necessarily not a nursery. I think, so leopard sharks and bat rays are still believed to be using the slough as a nursery. I caught a lot of immature bat rays, and so I think this is still the case. I just didn't catch like the right out of the stomach ones. The leopard sharks, I think it's overall just harder to catch some of them. It might be that they're using, so like I didn't sample in the lower slough at all, and there's some eelgrass habitat over there. So it could be that some of the animals are hanging out over there, and I wasn't able to sample it. It was just that for the thornback rays, so they're a little bit more similar to the round stingrays, and I thought so it was possible that they're only using the slough as a gestation area, and they did seem to leave the slough earlier than the other species. It might just be that they're more susceptible to the temperature changes. And there was a little bit more rain that year, so it would be interesting to see if this was different on a drier year.
I got a question. Did you talk to anybody that back in the day when you used to be able to use different gears? Did you ever talk to anybody that did any sampling with like seine nets out there that may have caught like newborn leopard sharks or anything?
I know from the derby data that there are small animals that were caught, like the neonates. I also know that Larry Tallant's study, he used 3-inch mesh on his gill nets, and he caught a lot of the smaller ones as well. But I don't know about the seining.
Greg might be somebody you might know, or even Jim who was around in there back in the time, because Greg used to do a lot of seine, have his class do seine others. You might just check with him if you haven't asked him that, if he's ever, any of his recollections, they ever caught some.
Well, I know from like Aaron Carlisle, I'm sure he talked to Greg, and he wrote the Derby paper. He was saying that nobody actually knows where any of them are pupping. So some were caught, but I mean, it's hard to observe, and yeah, we just don't know.
Any other questions? Wes isn't back with the food yet, so he has some questions.
You showed that video of the Thornback that was on the Longline with the two males biting onto it. Were you able to pull them all out, or did the males let go when you pulled them?
I did get the males. Well, one of them swam off. There were 3 initially on that animal. One of them swam away, like when I pulled it up. But yeah, just their tails are pretty easy to grab, so I just got him.